Okay, so my name is Pieter Colin from Groningen University, and it's an honor for me to moderate this third session of today, where we will talk a little bit more about methodological aspects of the work that we do and the research that we do. So it is my pleasure to announce Dr. Alex Proekt. Dr. Proekt is an associate professor of anesthesiology at the University of Pennsylvania. And Dr. Proekt majored in biological sciences at Carnegie Mellon and obtained his MD and neuroscience PhD from Mount Sinai School of Medicine. After passage at Weill Cornell Medicine, where Dr. Proekt made it to faculty, he moved to the University of Pennsylvania in 2015. So Dr. Proekt has made considerable advances to our understanding of the anesthetized brain and how the brain transitions between different phases during into and out of unconsciousness. Today, Dr. Proekt is going to discuss the origins of anesthetic hysteresis, and I'm sure that this will inspire a very interesting discussion afterwards. So thank you, Dr. Proekt, the floor is yours.
Thank you very much. Is my mic working? Perfect. Thank you for this kind introduction. I'm really honored to be here, especially since I'm not really a pharmacologist. I'm more of a neuroscientist, so it's great to have our work raise interest in the pharmacology community. So I'm going to get right to it, if I can advance the slide. Okay, there we go. So what is anesthetic hysteresis? And this is the kind of phenomena that has been really emphasized by the work of my good friend and colleague, Max Gelbs. And basically, it points to the fact that if you compute, how do I point with this thing? Yeah, there you go. If you compute the dose response curve for induction, you get this right shifted one. And if you compute it for emergence, you get a left shifted one. And Max has demonstrated this in a number of species with a number of drugs. He mutated genes and proteins to show that he can affect it. But in and of itself, hysteresis shouldn't be surprising. It's a very common phenomenon in nature. Essentially, the only thing you need to make hysteresis happen is to have a delay between the output, in this case, the state of anesthesia, and the input, in this case, the drug concentration. So I'm really having a hard time. Oh, okay, all right. Oh. So in fact, there is a very well-known and very well-understood explanation for why there should be hysteresis, and that comes from the standard PKPD models. And I should not be the one telling you guys about this because a lot of you have worked and built these things. But it basically says that, of course, there should be a delay between the state of anesthesia and the drug concentration, and that is because you're not giving the drug directly at the place where it acts. The drug needs to get to its site of action, which in PKPD verbiage is the effect site. Okay, and just to be very clear, I think that's absolutely true. There is simply no reason to doubt that at all. But PKPD models go a little bit further, and they say that is the only thing that is going on. That is, if I knew the concentration at the effect site, there would, in fact, would be no hysteresis. There would be just one dose response curve. Okay, and it is this part of the model that I think maybe is beginning to be challenged a little bit by some of the results that we and others have provided. So just to make it painfully explicit, if I knew the effect site concentration, I would expect, in this case, 50% of the effect. And if we make this into a more real world scenario where there will always be some errors in concentration measurements, some individual differences, then I might expect to have a distribution of responses. And nevertheless, this distribution will be centered around the true effect. But what I will try to convince you of is that this is the part that is a little bit different from what we observe. In fact, what I will try to show to you is that at many different anesthetic concentrations, the distribution of states of the system has multiple peaks. Here, for the sake of example, I'm showing 2. There will in general be more, but 2 is enough. And it turns out that this has some serious consequences. That is, for instance, this kind of observation suggests that the system will stochastically switch between different states. Maybe if I could ask you to advance. Thank you. The system will resist state transitions. Next, please. this resistance to state transitions is sufficient to give rise to hysteresis that is independent of PK. That is not to say that PK doesn't contribute to hysteresis, it absolutely does, but there are some additional factors that are not a part of the standard PK/PD model. Next, please. And but more interestingly than that, again, hysteresis is sort of a very generic phenomenon, but I do think that some of the results that we and others have been getting tell us something about how the drugs actually work on the brain. Next, please. Well, let me just begin with observations, okay? So what you're seeing here is a local field potential. You can think of it as the same way as you think of an EEG. This happens to be the one in a rat. I'm sorry, black backgrounds usually work in darker rooms, but I hope you can see that there is a white part of the trace followed by a red part of the trace. And you can see that the character of the oscillation changes abruptly. Okay, just to show you how abrupt it is, I've zoomed in on a 5-second chunk of data. Okay, and you can see that there is an abrupt change in the oscillation seen in the local field potential. But the kicker here is that we've done nothing to make this happen. Okay, this is a rat that has been anesthetized at 1% isoflurane for hours. There is no surgery. There is no stimulus that we are delivering to the animal. So we've seen this quite a lot. And almost 10 years ago now, next piece, we characterized the nature of these fluctuations by putting electrodes in the cortex and the thalamus. We've also done these recordings in the brainstem. I won't have a chance to tell you about them. Next, please. And what you're seeing here is a power spectrogram of these fluctuations, because you can see that this experiment takes a really long time. This is time in hours. And all I really want to show you is that, look, at every single concentration, brain appears to fluctuate between several discrete activity patterns, where the duration of each pattern is a lot longer than the time it takes them to switch from one pattern to the other. Next, please. So we want to study the distribution of these activity patterns, but it's a little bit unwieldy to work with the spectra. So we did sort of a pretty straightforward dimensionality reduction technique, principal components analysis, and you'll see I can find 2 projections of the data that cover about 60% of the variance, and in 3, I get about 70. Next, please. So now I can plot for you the distribution, the histogram of the spectra projected on the simplified way. And as I promised you, at every single anesthetic concentration, there's more than one peak in the histogram, and the regions between peaks are separated by areas that are very sparsely populated. If I repeat this analysis now in 3 dimensions, I get these blobs known as clusters, and we've done this up to 5 dimensions, and you get pretty much the same results. It's just a lot harder to show these things graphically. Next, please. Okay, and since this time, several other groups have shown similar results. This, for instance, is work by Yumi Ishizawa. So Yumi has shown this from Mass General. Yumi has shown this in non-human primates. And she used a variety of anesthetics. The data on the left comes from propofol experiments. She's done similar experiments with dexmedetomidine. And in this case, she's not looking at the spectra, but at the coherence. But again, I hope you can see that there are abrupt transitions. And I also see that I had too much coffee, and the pointer jiggles a little bit too much. On the right is work from Tony Huditz's lab from Michigan, where Tony recorded firing of individual neurons. In this case, it's rats. And in this case, it's desflurane. And again, statistics of this firing are form discrete groups as clusters. And he has shown essentially very similar things now with a different anesthetic and across a different scale. Next, please. So, but I mean, so I think this is true. I'm pretty sure that if you did this experiment, you would observe the same thing. But you know, are these results unassailable? Could one object to these findings? And of course you can. Sort of an obvious thing is that, well, I'm trying to keep the concentration constant, but how do I actually know that it is? Okay. And another objection is I'm trying to not give the animal any stimuli, but how do I know there aren't any stimuli from the environment? And of course, next, please. Of course, I can't really directly rule these out. I can't measure the drug concentration in the brain at the same time as I'm measuring brain activity. You know, I cannot do these experiments at 0 Kelvin, so I cannot rule out the possibility that some stimulus is triggering these things. But next slide, please. One thing we thought about is that, well, say for instance, I was wrong. And say for instance, anesthetic concentration fluctuations were driving fluctuations in brain activity. Next, please. In this case, I would expect that many parts of the brain will switch their state at the same time. But this is an example, simultaneous recording in the motor and the visual cortex of a rat against steady state 1% isoflurane. I hope you can clearly see that something very dramatic happens in the motor cortex and nothing much happens in the visual cortex at the same time. So this gave us an idea that maybe not all parts of the brain switch their state at the same time. So we thought we should quantify this. Next, please. But before I tell you about these results, I just want to put down a couple of basic facts about the cortex in mammals. Specifically, the cortex is made up of 6 layers, and these layers do slightly different things. The important bit here is that layer 4 of the sensory cortices, and in this case, we'll be talking about visual cortex, is the layer that receives the input. Okay, next, please. And I want to also say a couple of words about the scale, spatial scale in which we're talking about. So the thickness of the cortex in a rat is about 1.5 millimeters, so pretty small distance. So the different layers are roughly on the scale of a couple of 100 microns in width. And we have electrode arrays that we can implant through the layers of the cortex and simultaneously record activity in all cortical layers. And we can place multiple such electrodes into a single animal. Next, please. So it's just to prove to you that indeed we can get these electrodes into the cortex and the visual and motor cortex. You can take my word for this. Next, please. So then, using the methodology not entirely dissimilar to what we did almost 10 years ago, we took each spectrogram for each channel and we clustered it. And just like before, we found distinct clusters. So now the top several channels are the different layers of the motor cortex. The bottom several channels are different layers of the visual cortex. And you can see that sometimes a lot of channels change their state at the same time. But definitely much more commonly than that, you only see 1 or 2 channels switching their states, while the other ones do not. So we thought we should quantify this propensity of 2 channels to change their state at the same time. Next, please. And we did this using standard information theory measures, specifically mutual information. So this mutual information measure is normalized such that if 2 channels were always switching at the same time, the value would be 1. So we do find that if 2 channels are in the same cortical regions, just in different layers, there's more mutual information between them than between channels that are located in different cortical regions. That being said, the median mutual information is only about 0.3, which means only about 15% of the variance of one channel can be explained by another channel within a couple of 100 microns. Next, please. So then we could also use this setup to ask about the role of the potential role of the stimulus. So if the stimulus, specifically in this case, the visual stimulus, because we're recording in the primary visual cortex, was driving a switch in the state, then we would expect that the layer 4 will be particularly strongly coupled to other layers. But if it had nothing to do with the stimulus at all, we shouldn't expect any preferential involvement of layer 4. And in fact, what we found is that layer 4 is much less closely coupled to other cortical layers than other cortical layers are to each other. Next, please. So I just want to sort of summarize data from a variety of systems. I've shown you data from rats and non-human primates. There's also data in the mice that show similar things, data from different anesthetic agents that all converge on the same fact that under a fixed anesthetic concentration, brain activity fluctuates between several discrete states. And there is no real plausible way, at least in my mind, how I could get an anesthetic concentration in the brain to fluctuate on a scale of 200 microns. That doesn't seem very likely to me. Next, please. So to me, this is fairly, a combination of all this evidence is fairly conclusive in that knowing the drug concentration alone is not sufficient to specify the state of the brain unequivocally, right? Next, please. So, and you might wonder, what on earth does this have to do with hysteresis? This, of course, is Boltzmann. Okay, and we used this equation right here to compute mutual information. This is Boltzmann entropy. But for the next part, we'll need his other equation. Next, please. And that's the Boltzmann equation, which basically says that the probability of observing a state, a particular state of the system, is related to the energy of that state. Next, please. So basically, what I tried to show you is the distribution of states of the system has multiple peaks, more than 2 generally, but again, 2 is enough. So what that means is that the energy landscape that gave rise to this distribution of states must have multiple wells. There is one well for each peak in the distribution. And because the distribution has a trough, that must mean that there's an energy peak between them. Okay, next please. Right, well, that's fine. But the Boltzmann distribution only tells you what happens at steady state, and hysteresis is inherently a dynamic phenomenon. So we need help from 2 other physicists, Adrian Fokker, Max Planck. This is the Fokker-Planck equation, which basically says that if you place a system on some part of this energy landscape, how is it going to evolve over time? And it's going to evolve based on two forces. One is drift, right, which means it will always roll towards the local energy minimum. It's always going to roll downhill. But there is another variable in here, and that's diffusion, right, which is a random motion. So in the absence of any diffusion, the system would roll down to the bottom of the wall and stay there forever. But with random motion, it might jiggle about, okay? So that means that there are two components here that we'll be dealing with. So next, please. So let me relate this to anesthesia, right? So again, just to remind you, I've shown you that a distribution of states has multiple peaks. For the sake of this little model, I'm only going to consider two, but none of what I say depends on this. So if I say if your concentration is very, very low, only the awake state is possible. If it is very, very high, only the anesthetized state is possible. And in the middle, both states have some degree of stability. So if I calculate what should happen to the system without noise, I get these curves, which clearly describe hysteresis. Next, please. Now let's imagine what would happen if I dialed up my anesthetic concentration to EC50 and left it there. Next. If I waited a little bit, the system will stay right where it is. But if I wait longer and longer and longer and longer, eventually over some time course is going to approximate the Boltzmann distribution. And how quickly is it going to approximate the Boltzmann distribution? Well, that depends on the noise. If the noise is very little, the system would be very unlikely to hop across the boundary. If the noise is a lot, the system is going to explore the space very quickly and approximate the Boltzmann distribution quickly. Next, please. And indeed, in simulations, we have shown that hysteresis will depend on the amount of noise and should collapse on a certain time scale. It has nothing to do with PK. And unfortunately, I don't have time to show you some of the data, but we have some, albeit preliminary evidence at this point to show that this exact thing happens in behavior of mice. Next, please. I think when we first started coming across this, this was met with bewilderment because it seems strange to think that when you fix all the influences, in this case, drug concentration, why would the system be switching between several states? That seems counterintuitive. But in fact, it's a very generic property of complex systems. Just to show you what that means is that I made a little network of two neurons. Note my disclaimer. I don't think this is the brain. This is just a toy system, right? And let's say, I imagine that my anesthetics act on this synapse, okay? When there is little anesthesia, the synapse is weak, but anesthetics say potentiated, like for instance, for some of the mechanisms that people have spoken earlier about, for instance, through potentiation of GABA transmission. So this is an inhibitory synapse. And you really can't see these red traces. I'm really sorry about that. I don't know if the lights can be dimmed, but in any case, you have to take my word for this, is that if the anesthetic concentration is low, this kind of system will exhibit irregular fluctuations of low amplitude, like what we see in the awake brain. If the concentration is high, then it will exhibit high amplitude, low frequency oscillations, just like we observe in the anesthetized brain. And in the middle, it will switch. And this is an example of the switching between different characters of oscillations. And if I add a lot of noise, the system will switch all the time. If I add a little bit of noise, it won't. So in fact, having more than one stable configuration is a generic property of nonlinear dynamical systems. And the brain, of course, is a nonlinear dynamical system. Next, please. Just to show you that this is not just some mathematical fantasy. This is a recording of a single neuron. This is not a model. This is a neuron from aplesia, a system I studied in my PhD, where this neuron is exhibiting bursting behavior. And then a little blip arrives. And you can see that the neuron switches now into firing tonically. And that tonic firing is sustained for quite a long time until the system decays back to its original state. This, in fact, is very, very similar to what happens in the thalamic neurons as we go between states of sleep, or state of wake, or state of anesthesia, state of wakefulness. Next, please. Okay, I've told you a lot about different kinds of brain squiggles and how they flip and flop, and that could be very interesting. But ultimately, of course, I anesthetize people. I want to know, are they awake or are they not awake? So does this have any bearing on that? Next, please. Right, so I work with mice mostly, and rats and other critters. So we can't really ask them if they're awake. So we use a very simple behavior, and it has many limitations, which I will freely admit. But basically, and this has been done for many, many years, you put a mouse on its back, and you see if it is able to right itself. And many people have worked on this reflex to show that roughly speaking, EC50 for this reflux closely parallels MAC-awake in humans for a variety of anesthetics. But nevertheless, this is our measure. So let's see what should happen at EC50. Well, at EC50, I should then expect one mouse to be able to write itself and another mouse fail to do so. Next, please. The key prediction is if I repeated this experiment, I better get the same outcome. Okay, next, please. All right, but this is actually what happens. These are 20 mice that have been placed in anesthetic chambers for 2 hours prior to the beginning of this experiment. And now we tested their writing reflex 40 times every 3 seconds. And you can see that every single mouse, without any exception, sometimes responds and sometimes does not respond. We've now done this on hundreds of mice. This is always true. Next, please. So there could be another explanation for this. Well, maybe at EC50, every mouse responds to 50% of the stimuli, which is akin to tossing a coin. And if the coin lands on heads, say you are able to respond, and otherwise you can't. But the key proposition here is that the probability of response should be the same on every trial. Next, please. But this is not the case. So if I look at the probability of failing to respond for a mouse that responded on the previous trial, I get a very different probability from the same mouse when it had failed to respond in the previous trial. That is, the fluctuations in behavioral responsiveness also show this kind of inertia that I've shown you with brain activity. And this kind of a state dependence or history dependence, again, is just simply not a feature of a standard sort of understanding of how these drugs work. Now, you might say, well, maybe this flipping and flopping is not really, it's maybe noise related to the assay itself and things of that nature, in which case we shouldn't be able to modify this tendency to resist state transitions. Next, please. But it turns out that you can, and one of the simplest way of showing it is to use different anesthetics. So here we titrated iso, sevo, and halothane such that at the level of a population, they give rise to the same overall probability of successfully responding to the stimuli. You can see all of these are done after 2 hours of equilibration, so we're at steady state, and you can see that at the level of population, there is absolutely no change in response probability. But if you look at these data for a second, you will see that the patches of blue and green are clustered in the halothane data relative to isoporine data, and we can quantify that. Next, please. And you can see at the level of probability of writing, the 3 drugs are not statistically different, but at the level of tendency to resist state transitions, there are huge and consistent differences between these drugs. So we can modulate not just the potency of the drug, but how sticky of a state does it elicit. Next, please. So I want to put these results into a bit of a context, I think, which maybe will help in this sort of resolve some of the maybe misunderstandings or issues between this approach that I guess I'm advocating and the standard PKPD approach. Next, please. So at the dawn of pharmacology, this is Langley, right? He deduced the existence of a receptor in a very abstract way. This was based on very clever experiments at neuromuscular junctions and things of that nature. So it was brilliant, right? But he had no idea what receptors actually are. Next, please. And following up on this work, several brilliant people have suggested that perhaps it is the drug adsorbing to the receptor that explains the existence of the dose-response curve. Next, please. But there became to be some difficulties. For instance, and we have seen some of the examples of this kind of surprise this worked out, that not all drugs elicit maximal response at saturating concentration. We call these drugs, partial agonists or whatnot. Next, please. But it was impossible to understand how these drugs work until we had an understanding of what receptors actually are. And receptors are complex macromolecules that have their own complex dynamics that help explain why certain drugs are full agonists, antagonists, or partial agonists. So for instance, this is a recording of GABA-elicited currents in a whole cell that has many copies of GABA receptor. And this is recording of a single receptor molecule. And you can see a very parallel structure to what I've been trying to tell you. At the level of a population, there's a smooth dose-response curve. At the level of a single receptor, the receptor fluctuates between sometimes conducting current and sometimes not conducting current. And these states have certain characteristic timescales, meaning once a state is established, it tends to last a certain amount of time. Next one, please. And this is just sort of to drive what I'm trying to say home, is that we can certainly use those response curves and things of that nature to describe what we observe at the level of phenomenology. But if we really want to try to understand how the drugs work, we have to understand what they do to an individual receptor. And for instance, Alex and Douglas have spoken a lot about this. This is not a simple proposition at all, but without that, we don't have much in the way of understanding. Next, please. Great. So I guess what I'm trying to say is that it's not that there is a conflict or any disagreement between a standard PK/PD approach and what we've been trying to do. It's just simply an acknowledgement of the fact, next please, that when the drugs act, even if we knew how they acted at a molecular level, at the level of a receptor, these receptors are components of a vastly complex system. And changing one component of the system can affect its dynamics in all sorts of complicated and maybe sometimes counterintuitive ways. But without understanding that, I don't think we have much of in the way of hope of understanding what anesthetics do to the brain. Next, please. And this is basically to say that this kind of a distinction between the nature of phenomena that are observed at different scales is certainly not unique to pharmacology. In fact, this is a very prominent feature of physics. Of course, phenomena on the quantum scale are discrete, whereas phenomena on a macroscopic scale are continuous. So that's all I have to say. Thank you guys very much.